much, Kayla, and warm greetings to everyone. Thank you all so much for taking the time to join us for the July GFRA webinar. Today, I have Dr. Mariana Perez as my co-panelist assisting me with the webinar. And together, on behalf of the GFRA, we are very excited to have actually our GFRA Exco Financial Director presenting the webinar today. And it's on a very hot topic of FMD for 2025, which was the FMD outbreak in Germany. So we're very excited to have him. And for those of you who do not know, I'm talking about Dr. Michael Eschbaumer, and it is my pleasure to introduce him to you today. He graduated from the Faculty of Veterinary Medicine at Ludwig Maximilians University of Munich in 2008. And after earning his doctorate at the Friedrich Loeffler Institute, which is the FLI, the German Federal Research Institute for Animal Health on the island of Reims, he worked as a postdoctoral research at the University of Calgary, Canada. And he also worked at the Plum Island Animal Disease Center in Orient, New York, USA. Since 2016, Dr. Eschbaumer has been the head of the German National Reference Lab for FMD. His research interests include safe and effective FMD vaccines, persistent FMD infections in ruminants, intra- and interspecies recombination of aptoviruses in vitro and in vivo, and approaches to improve biosafety in the collection, transport, and testing of FMD samples. So Michael, welcome. We are very excited to have you and we look forward to your presentation. So I hand over to you.
Thank you, Melanie. Thank you for the kind introduction and for the opportunity to be here today. Yes, you've called it a hot topic, and indeed, I mean, I've been asked to speak about this at various venues, various situations in the past. So I apologize if there are any people in the audience who've heard this talk before. I always try to bring in some new things, and also this time we're going to have some pictures that at least I haven't shown before. But if it becomes too repetitive, I mean, feel free to go off and do something else with your morning or afternoon. You have been warned. Anyway, the National Reference Lab has an important role in an outbreak, but it's not the only important player. There are, of course, many other people, both at the federal level, at the state level, at the local level, that contribute to detecting an outbreak, controlling an outbreak, and the aftermath. And I try to bring in their perspectives as well as we go along. Some of them are mentioned on this slide here, but obviously there are many more. So the situation in Germany before this year has been very quiet with regards to FMD. The last FMD outbreak was way back in the 1980s. The last big outbreak in Europe, obviously, was the 2001 situation in the UK, the Netherlands, France, and the Republic of Ireland. There was this smaller scale accidental release of FMDV from a containment facility in the UK in 2007, and then the last outbreak in the EU, 2010. In Bulgaria. And in the meantime, we've really gotten used to this map that you see here on the right, with like all of Europe free of FMD. But at the same time, obviously, FMD has always been on our minds. FMD is an important disease, is a global disease. Before we go into this outbreak a little bit more, I just wanted to remind you of some concepts with regard to FMD that may be relevant for understanding like what we have done in response to the outbreak, and some of the thoughts that we have had in analyzing it after the fact. So of course we have ruminants and pigs as the susceptible animals, and then it's often said that pigs maybe are more susceptible to oral infection with FMDV, for example, by contaminated feed or untreated food waste that is being fed to them. At the same time, maybe airborne infection is not as big of an issue for pigs. Whereas an infected pig will produce a lot of virus, shed a lot of virus, and can then in turn infect, for example, ruminants by air. Obviously, the most efficient method of FMDV transmission certainly is the direct contact between an infected animal and a naive animal. Small ruminants, maybe they're susceptible, they will get infected, they will shed virus. Clinical signs sometimes are severe. I mean, it's certainly not always an inapparent the disease in small ruminants, but maybe less consistently so than with large ruminants or pigs. And then on the other side of the screen here, the other corner, we have the wild animals. And at least as far as Europe is concerned, I think there's big doubts as to whether wild animals really play a big role in the epidemiology of FMD. We know that they're susceptible, they can be experimentally infected, they do get sick to a variable degree, they do shed at least some amounts of virus, and at least in the outbreak in Bulgaria, they were involved in the outbreak in the sense that some were found to be virus positive, more were found to be serologically positive. But whether they actually were an active driver of the outbreak, I think has not been completely resolved. And similarly, we have reports from Israel where also wild boar are infected, are found to be infected, but whether like the infection goes from the wild boar to the domestic animals or the other way around or both is still a bit up in the air. So definitely there needs to be a question. Anyway, so this is a slide that was generously given to me by Don King of the World Ref Lab for FMD. He has this in many of his presentations, just trying to give an overview of the global situation of FMD. I'm not going to go in detail because we're really just going to talk about Germany. It's just to emphasize that it's always a dynamic situation. I mean, there's always FMD all over the world, and even during this period of peace that I mentioned earlier, where in Europe, like there was no FMD that we had immediately to deal with. It obviously was always all around us, very much so. As you can see here as well, with a lot of diversity in terms of circulating strains, both in North Africa and in Western Asia as well. And this has always been a concern for people in the FMD field in Europe, that the viruses they're really not that far away, but is a virus going to be introduced? How is it going to be introduced? And if so, is it actually going to come from the areas that we think most likely, or from an entirely unexpected direction? Who knows? And it's always kind of like a bit of a crystal ball exercise, trying to predict what actually is going to happen, and then often you get it wrong. So in this particular instance, what have we been dealing with? So our story begins on the first day of the year 2025. 14 water buffalo, a fairly small herd that were being kept on a large pasture just outside of Berlin, in the northeast of Berlin. I'm going to show you a map later. A very extensive operation. So these animals were just out in the pasture. I mean, they were visited every day. They were given feed, but not much was going on with them usually. So over New Year's, the owner actually wasn't there. New Year's Day, a friend of the owners was taking care of the animals, and they noticed something wasn't quite right. These animals weren't the same, weren't behaving the same way as usual. And the owners came back and they found they're not eating well. Like there was this one cow, a favorite of the owners, who would always come over for some sugar beets, and she wasn't showing up. And then they observed lameness in at least one animal in the herd as well. So something is off, but nobody really can put a finger on it yet. What is going on with these animals? And then, shockingly enough, the next day, two of the adult buffalo, two females, were found dead. This is an actual picture taken on the farm that was given to me by the state animal disease colleagues in Brandenburg. And I mean, that of course really spooked the owner. So they were just really concerned about their animals, really wanting to do something. And then the following day, a third animal of the herd was found dead, another adult female. And then the owner started like, I don't know, searching the internet, what's going on with animals, trying to get a hold of their veterinarian, calling other people who own buffalo. I mean, there's a surprising number of these people apparently in Germany, which I didn't know before that outbreak either. And apparently, the one of his friends in the buffalo industry suggested, well, just take the animal, take it to the lab, get a post-mortem examination done, and figure out what's going on. And that's what the owner did. So he had some farm equipment on the farm. He put the dead animal. I mean, we're talking several hundred kilos worth of carcass here, but he put it on the bed of his pickup truck and went down the highway to the regional veterinary laboratory to have the animal tested. The animal went to necropsy the following day, January 7th. We're going to talk about the necropsy findings in a bit. But the suspicion at that moment, from what I've been told, went more in the direction of maybe a case of blue tongue disease, which has recently been on the rise again in Germany. While this is going on back home on the farm, the feed uptake of these animals has returned to normal. They seem to be doing better, and so the owner just gives a call to the local rendering plant to please come and pick up these other two carcasses. So they have been picked up, disposed of. We do not have any samples of these first two dead animals that you see in the picture here. We really don't know more about them other than that they're dead. Anyway, so at the regional lab, in the meantime, they tested for blue tongue virus. It was negative. They tested for a whole bunch of other diseases, both viral and bacterial, and nothing really came up positive. And then as part of the workup, they also tried to rule out foot and mouth disease by real-time PCR. Unfortunately, that didn't go as expected. And on January 9th, which is Thursday, they found that the samples from this animal that underwent a post-mortem examination actually were so some of them were quite positive for foot and mouth disease in the PCR. So then they gave me a call in the afternoon and said, hey, we have a situation here. We're gonna send some samples here. So usually it's not all that far from the regional lab to FLI. They called me around 4 in the afternoon and said, oh, delivery of the samples will be around 8, between 7 and 8. So we got everything ready. We got all the people ready to go for the samples, but the samples didn't come. We were just sitting here, obviously making lots of phone calls, sending lots of emails, just getting everybody really amped up, but there were no samples. At this day, there was a huge snowstorm in the Berlin and Brandenburg area, and the driver was kind of inching along the highway at walking speed, just trying to get the samples to us. They finally arrived just before 1 a.m. Friday night. And of course, we immediately went to work. Within a couple hours. Unfortunately, we had to fully confirm the results of the regional lab and notified all the relevant stakeholders on the national and international level, and began with virus isolation and virus characterization. We found that the original material we had was not positive enough to allow serotyping by antigen ELISA, so we had to wait for the virus isolate. But in the late hours, Friday afternoon, it already started to look as if the virus isolation had taken hold. I started to see the first little indications of CPE, so I was quite hopeful that would work out. I'm going to show you a picture of that as well. And then Saturday morning, we had a strong CPE, we had a virus isolate, and we also had preliminary results from sequencing rolling in. And over the course of the day, we could then both from the isolate confirm the serotype to be serotype O, and we got sequence information, not just BP1 actually, but the time 2:30 Saturday, we had the whole genome sequence, and we could positively identify this to belong to the OMISA SA 2018 lineage, which we then also communicated. This is just the real-time PCR results of these samples from the buffalo. You see the lung is most highly positive. That's the material that I used for the cell culture, and that's what I saw Friday afternoon. So I already thought, okay, this is going to be good. Then Saturday morning, full CPE, and that material then also was strongly positive in the antigen ELISA, confirming the serotype O with the usual cross reactions with the other Eurasian serotypes. Nothing to worry about, really. I think this is very indicative here. Anyway, so this is the necropsy report of that third dead buffalo. Big gal, as I said. We do not have any pictures. Unfortunately, the pathologist who was doing the postmortem did not take any pictures. But if you look at the report, you see that, oh, there was a third of the tongue epithelium missing from that animal. There were other ulcerations on the rumen mucosa. There were no lesions on the feet observed in the PM, but so there was a severe congestive hyperemia of the lungs, like indicative of cardiovascular failure, and also in the histopathological examination, they did find some signs of myocardial necrosis as well. These samples were then passed on to the pathologist at FLI and tested again here a little bit later, and there they confirmed that there was FMDV antigen in the lungs and the myocardium associated with the visible lesions. And as I showed you, the lung tissue was also highly positive in the PCR. So taken together, for this animal, I would definitely say that the animal died of acute FMD. And there was no detectable FMD antibody in the blood of this one animal. So this was a more recent event for this one. This is not a picture of this animal. It's just a picture that I found in the excellent online lesion library of UFMD, just to give you an idea of what 30% of the tongue epithelium missing may look like in an acute case. I found this picture to be quite impressive. Please do go and check out this lesion library. It's great. There's a lot of really good pictures in there. So we confirmed the outbreak on January 10th, Friday. On the same day, the state authorities in Brandenburg got to work. They brought in like an external contractor to kill the buffalo. They went and took samples, of course, and they also decided to cull animals on 3 nearby farms close to the outbreak farm. 261 susceptible animals in total, the majority of which, like 250-ish, were pigs kept at one organic style farm just a little ways down the road, and the rest were just ruminants, a cow or two, and small ruminants kept at 2 other farms. And then over the course of the epidemiological investigation, they also identified a high-risk contact a little bit further afield that had received animal feed from the outbreak farm, and that was culled the following Monday, January 13th. Here are the zones that were then declared based on EU legislation. So this is the city of Berlin. Berlin city limits are this purple line here. This is the 3 kilometer zone and 10 kilometer zone. 3/4 of the zones are on the territory of the state of Brandenburg, and about 1/4 is on actually the territory of the city-state of Berlin. These are some pictures that were also very kindly given to me from the colleagues in Brandenburg, what they found when they arrived at the farm on Friday morning. Remember, this is like, yeah, middle of winter. in the northern hemisphere, and there was a bit of a snowstorm going on the night before. So if you can't actually see the buffalo, they're back here. So this is really a huge area, about 100,000 square meters overall, with a little lake, and you can see kind of like this rolling terrain. Before this became a buffalo farm, this was supposed to be a golf course, but that either failed very quickly or never really materialized. So the landscaping was done for the golf course, but then they ended up putting the buffalo on this golf course. If you look very closely, you see something here, top right. Maybe that's saliva, maybe that's just snow. I don't really know, but it could also be FMD related. These are the buffalo here. They were being shot to kill them, and then were transported from the site where they were being killed to the site where they were being sampled. This is the colleagues at work here trying to take samples. And I mean, you just see the environment that they had to work in. Animal disease control is never fun, but a situation like this, I think, makes it even worse for everybody involved. But they did a great job. It all went without a hitch very quickly. On the Friday, the situation was brought under control on that farm. Before we go into the lab results that we got from these buffalo, just a quick reminder kind of the diagnostic parameters of FMDV infection. This is another great slide from the Pirbright Institute. Obviously, you get the first virus shed in saliva and nasal fluid, just matching at the sites of primary replication. This begins before clinical signs. This begins before viremia, and this lasts longer than viremia as well. And usually, I mean, this is all very, very abstract, obviously, but in general, I would expect the swabs to contain more virus or more viral RNA by and large than the blood or the serum would. And then, of course, if you have the acute disease, you have the vesicular lesions, which then would contain the highest amount of virus when they appear. And then after maybe a week, give or take, certainly with differences, different species, etc., you do have the antibodies rising in the blood, and at the same time, the antibody response will terminate the viremia. So it's very rare that you actually find animals that are both positive for viral RNA in the blood and positive for specific antibodies with FMD. With other diseases, say blue tongue virus infection, it's completely different, where you can have these long periods of time where there's both an antibody response and detectable viral RNA in the blood. And then, of course, if you have persistent infection, that will go on even longer. Keeping that in mind, let's have a look at the lab results that we got from the samples collected from the 11 buffalo that remained on the farm when the disease control people arrived there. So unlike the animal that had dropped dead and was tested at the regional lab, or was necropsied at the regional lab, these animals all had detectable FMDV antibody. They were all antibody positive to varying degrees. And at the same time, they were all, with the exception of this one animal, still positive for viral RNA in nasal swabs. Some of them also in blood, but just with very high CTs, very low levels of viral RNA, which matches what I showed you on the previous slide. There was some detectable viral RNA in the mouth lesions as well, but as you can see from the pictures, these lesions were already quite old. So in these animals, we're already like talking close to a week, in some cases even more, after probably the time these lesions first appeared. And the only lesions that were still highly positive were the foot lesions. I have limited experience with that myself, but I've been told that this is not too unusual, that often, like, just due to the higher turnover of mucosal surfaces and the saliva, etc., mouth lesions do become PCR negative quicker than foot lesions. Foot lesions often remain at higher levels longer, at least. That's what I've been told. So all seems to match, but all seems to be indicative of, yes, a little bit of an older disease situation in these animals. Certainly not several weeks, but a week. So then what happened? Well, the usual, I mean, these animals were called, the others were called, there were movement bans initiated, and obviously a big epidemiological investigation, trying to find out where the virus came from, how it got into the farm, and then where it potentially spread from the farm, and also trying to establish a timeline. The investigation, one of the things that they found was that in early December, there had been one buffalo calf from the herd had been sold for slaughter, and they did manage to find some frozen meat of that animal somewhere. And we got sent that meat for testing, but there was no viral RNA or antibody detectable in there. So taken together, like what the owners told the investigators, the lab results, the clinical picture, etc. The conclusion was that the virus was most likely introduced to the herd in the second half of December, probably like closer really to the end of the year. But the route of introduction at the time remained unresolved, and as you're going to see, actually kind of remains unresolved to this day. This is an overview of all the farms involved in this outbreak. You see the yellow dot in the middle, that is the actual outbreak farm. Then the purple dots are the 3 depopulated farms. Then the very few red circles are all the farms in the 3-kilometer protection zone. Then you have the blue farms in the 10-kilometer surveillance zone. And this teal color here are contact farms outside of the zones. The contact down here in the southeast is the high-risk contact that actually ended up being culled. But the other 6 dots up here are farms that the rendering vehicle went to after picking up the 2 carcasses at the outbreak farm. They went to these 6 farms to also pick up carcasses, and this is how they ended up on the contact list. What you can also see, much to my surprise, is that there are actually a number of holdings of susceptible animals, so livestock, within the city of Berlin. And some of these were like actual, like, I don't know, petting zoo type situations where you would expect a goat or 2. But from what I've heard from the colleagues in Berlin who actually had to go and like take samples and investigate these other cases, some of these situations were really quite bizarre, like animals living in closest proximity to people, sometimes within their apartments. And obviously these people, by and large, really they did not care about FMD. They didn't much care for industrial agriculture, and whether or not Germany was FMD free as recognized by WOA or not certainly did not matter to these people at all. So they were not too interested in any sort of discomfort visited upon their animals in the course of the control of an FMD outbreak, to put it mildly. So where did the virus come from? We did obtain the full-length sequence, as I mentioned earlier, and together with colleagues at the European Reference Lab and at the World Reference Lab, we did find a fairly close match in the database. There is one isolate from the far eastern part of Turkey from late 2024 that has one nucleotide difference with the virus that we found in Brandenburg. It's important to keep in mind, though, that the sequence record is very sparse. I mean, we have this one isolate, then we have one earlier isolate from 2023 from Iran. It is also fairly close, even though not as close. While at the same time, we know that this particular virus or viruses of that lineage are also circulating in other countries in the region. And I really don't want to kind of punish the Turkish colleagues for actually taking the sample and putting the sequence online for us to compare to. There are other hotspots of this virus in the region. So just the general area, southern Caucasus, eastern Anatolia, northern Iran, northern Iraq, kind of is the geographical area where this virus likely came. This still doesn't answer the question how it got into the farm, and we're going to talk about that in a little bit. But before we get to that, just with the isolate that we obtained, we shared it with the European Ref Lab. We shared it also with Boehringer, the provider of the German FMD vaccine bank. And both of them shared their vaccine matching results with us. And as you can see, like all three available serotype O vaccine strains had fairly good heterologous titers, would probably have worked well to control this outbreak if it had been necessary to use the vaccine. Obviously, with the inactivation of the vaccine bank, you don't want to wait for the vaccine matching. So based on the sequence data alone, BI had recommended a bivalent vaccine containing the O1 Manissa and the Open-Asia 2 antigen. When Germany actually activated its vaccine bank on January 20th, mostly for cost reasons, they opted to get a monovalent vaccine with just, Pan-Asia 2. And then when the vaccine matching results came in, this was confirmed in the sense that we had the highest heterologous titers really seen for Open-Asia 2. And as far as I know, the Netherlands also activated their bank, and they also went for the monovalent Pan-Asia 2. That being said, in the end, we never used the vaccine in Germany, but it was available, it still is available, and it was then subsequently shared with Hungary and Slovakia for use in controlling their respective outbreaks, which were caused by a different virus, but a virus of the same serotype. So the serotype O vaccine also came in handy there. So talking about how did FMD get into this buffalo farm? As I said earlier, we don't really know. We just know a lot of possible routes of introduction that we ruled out. So there were no animals imported into the farm in a long time, and certainly not animals coming from areas where FMDV is endemic, or in this particular case, I mean the geographic area that I mentioned earlier. So this was a closed herd. They had one bull and a bunch of cows, and all the calves in the herd came from that bull. So there was no artificial insemination. There was no semen imported that could have contained any FMDV. There was also no feed or bedding or anything for these animals that was being purchased. The owner, so mostly they were on pasture, and they were fed some hay that came from pastures or fields immediately adjacent to where the animals were kept. Same for the bedding. All came from that farm, so no route of introduction there. The owner did not share any vehicles or equipment with anybody else. There were no like people who had immediate contact with these animals that had been recently traveled to areas where FMDV was endemic. And as you will see later, there was also like no indication of FMDV in wildlife around the site. So this is also not how FMDV got into these animals. Again, very different from some other diseases that Germany is dealing with right now, particularly African swine fever, where the migration of infected wildlife obviously does play a role, at least in some of the outbreaks that we've observed. So we rule out all this, and then like based on the Sherlock Holmes method of epidemiology, once you've ruled all these things out, whatever remains, as improbable as it may seem, really are the only things kind of still in play. And so we really have two big things here. Of course, one is the intentional introduction of the virus into the farm, be it by a disgruntled individual with some personal agenda, or some state actor in the sense of like hybrid warfare, state-sponsored terrorism, whatever you want to call it. Let me emphasize, we do not have any evidence pointing in that direction. It's just also something that we cannot rule out with the evidence and the observations that we have. And then finally, the accidental exposure to illegally imported food of animal origin that was contaminated with FMDV and somehow got to these animals. I have to highlight that this is again pure speculation. I don't have much in terms of evidence to confirm this, but we do know that some viable FMDV remains in certain kinds of dairy products, for example, like soft cheeses made from unpasteurized milk. It says here, I really thought this article was nice. Raw milk cheese is mostly safe, mostly. But not entirely, and not when it comes to FMDV. And so you have to know that the area where these animals were kept is close to a very busy spot for like outdoor activities. People from Berlin come to the outskirts of the city. They park their car and then they walk their dogs across the fields and into the forest, etc. And they walk right past the fence of this buffalo farm. So I don't know. Maybe somebody went somewhere over Christmas break and brought home some food, some raw milk cheese that may have contained virus. In the end, they didn't want to eat it or could. I don't know what happened, but anyway, the cheese didn't end up where it should. It somehow ended up with these buffalo who possibly consumed it. And as we said earlier, oral infection in ruminants is really not a big thing. So I know that this is not super convincing, but it's really it's all I've got. And one other story that's kind of that I'm trying to hint at here as well is that the owner during the epidemiological investigation said that they had seen crows kind of picking up trash, picking up stale bread, etc., in the area, and then dropping it into the water trough of the buffalo to soften it up so that they could eat it. And maybe it wasn't even people directly. Maybe birds were somehow involved in bringing the contaminated material to the buffalo. And unfortunately, we do know that there is a lot of illegal importation of food of animal origin into Germany, into the EU as a whole. We don't know how much, but it's thousands of tons. And there are these signs at the airports, where they tell you, hey, you can't really do that, but there's not a whole lot in terms of enforcement, and I'll get to that in a second. But I don't know, you know, maybe it was the birds who brought the cheese to the buffalo. Maybe it was somebody trying to get the buffalo's attention so that they could get a nice picture. I really don't. I'm grasping at straws here, as you can probably tell. What I do know is that there is not much enforcement of this ban on illegal importation of it's very different, say in Australia, New Zealand, North America, where you see these dogs like sniffing the luggage, etc. And I'm sure that's done here and there in Germany, but certainly not with the same intensity. And maybe that's something, if you want to take one thing away from this outbreak that you should do differently, maybe enforce the ban on illegal imports a little bit. I know it's very improbable, but if you try often enough, maybe at some point, even an improbable chain of events can actually come to pass. That being said, what happened next, or what else happened? I showed you the map with all the farms around, and there was like a lot of investigation in the zones. There were repeated clinical, serologic, and virological exams. All the farms were visited many times in the zones and the contexts. Roughly 10,000 animals almost overall. They were all negative. We found no FMDV antibody or a viral RNA in any animal other than these 11 buffalo that were still on the farm and the one that was brought to the regional lab. Nowhere. Then we looked at a lot of wildlife. I'm going to show you another slide on that in a second. Over 500 susceptible wildlife from the area were killed and tested, all negative. All animals that had been shipped from the region were negative. There was really outside of this one farm, we didn't find the virus anywhere. And this was then put in the appropriate reports, dossiers, what have you, submitted to WOA, and within a short time, in early March, containment zone was accepted by WOA, so that the rest of Germany outside of this containment zone that you see here then became immediately free of FMD again. So the FMD free status was reinstated. By the way, I don't know if you noticed that, but it's quite interesting to see animal disease control in Germany is like devolved to the States. So you have animal disease control Brandenburg, who does things a bit differently than Berlin. And so the zone boundary here ended up being drawn very differently from the part that is in Brandenburg and the part that is in Berlin. But anyway, it worked. So about a month after the containment zone had been installed and no new cases were found anywhere, this containment zone was also lifted by WOA, and about 3 months after killing and disposal of the initial herd had been finished, all of Germany regained its FMDV free status in mid-April. That being said, just because you're free again doesn't mean that trade immediately resumes at pre-outbreak levels. I mean, the people who deal with these sorts of things are still working on some of the third countries, trying to get them to accept German agriculture products again. So this is a small outbreak, right? I mean, it was one farm, but at the same time, with all the trade losses, etc., up to this point, I think the estimate of total economic damage caused by this outbreak is around €1 billion. So it's a lot. It's a lot of money that is destroyed when you have an outbreak, and it's gone with the first farm. So this is just one picture showing you how they were trying to kind of get an idea how much wildlife there actually was, how much samples they would need for a good to have a reasonable estimate of the prevalence. So the outbreak farm is somewhere around here, close to the A10 ring road, and in 2 nights they did a helicopter survey with like a thermographic vision and were counting the wildlife, and they found several hundred roe deer on both instances, and a roughly smaller number of wild boar. And I think we got a fair bit of these animals then actually shot and tested. Another interesting angle before I finish is the cleaning and disinfection. How do you clean and disinfect an entire farm? So we were dealing, or the colleagues in Brandenburg who gave me the slide, were dealing with 100,000 square meters of pasture land that these buffalo could roam free on. So any last little bit there could be contaminated. It was winter, so you didn't really want to count on the sun to get rid of it. So they fenced the whole thing off to kind of keep the wildlife out. And then they ended up dumping just 100 tons of quicklime on the whole thing, as you can see here, and then kind of till it to get it into the soil, spread it out as much as you can. And there was this lake, and they were having discussions what to do with the lake. Just dump quicklime in the lake or get the water out. But then what do you do with the water? So in the end, what was decided to also then put like a second fence around that lake and just leave it, let nature take care of it. What made this a bit more of a juicy issue was that at least one of the dead buffalo was actually found like on the shore of this lake where it had dropped dead. Anyway, so just to sum this up, it's a small outbreak, right? Still expensive, but it's a small outbreak. Why is that? It's not really what you expect from an FND outbreak. But I think we really got there early. We got it on the first farm. At the same time, the propensity to spread from this farm was low because it's still close to Berlin. There's some agricultural activity, but the livestock density there is far lower than it would be in other parts of Germany. You saw the map. I mean, it was a big zone with ultimately not that many farms, and a lot of them very small farms. And just by the way this outbreak farm operated with their animals on pasture, they didn't have many contacts. They didn't bring in animals. They didn't sell animals. Nobody came to pick up milk. Um, the people who had contact with these buffalo didn't go to other farms, etc. So just there was very little opportunity for the virus to actually spread from this herd. And with the small number of animals, there also wasn't like a big plume of viral material generated to be spread by the wind. And once it was recognized, the veterinary authorities really were on the ball and quickly and decisively put an end to the whole thing. And I mean, at the end of the day, sometimes you also just get lucky. And I think we really got lucky in this situation. But FMD is still a concern. And you saw just a couple of weeks later with the outbreak in Hungary and Slovakia, you always have to be on the ball. You have to be on the lookout for FMD. And there's just test as much as you can when you have any sort of weird goings on in terms of a vesicular or vesicular adjacent disease in susceptible animals. Get a sample to the lab, get it tested to make sure that you do not have FMD. That's it for me. Thank you very much for your attention, and I'm looking forward to your questions.
Thank you so much, Michael. Really, really amazing presentation. I love the way you did everything step by step and gave us such a nice story from beginning to end. I love your AI generated photos as well. Very nice touch to the presentation. Just a note to everyone that's present here today. Please, if you have any questions for Michael, please post it on the Q&A box, and then we will help get those questions to Michael. We have about 10 minutes to get through some questions, and we do have some for you, Michael. We will start with Alder Decker, and I think you kind of touched on this in your presentation, but he thanks you for a nice talk and the clear description of the case in Berlin. We see in the Netherlands that farmers sometimes feed bread from bakeries. If there is meatloaf, then the product could contain FMD virus. And his question is, do you know if the owner fed other foodstuff to them?
No, they did not get any food from any outside source. They got alfalfa hay from a field right next to the pasture, and they got some sugar beets that the owner also grew himself. No material from a bakery or any other like food operation.
Thanks, Mariano.
Yeah, thank you, Michael, for this wonderful presentation. Really was very instructive in terms of what happened there in Germany. I will take the privilege of being panelist, and I will make myself a question first. But it's a general question. How surprising was this outbreak situation for you as a scientist and as a part of a German animal sanitary system? How surprising was that? This was something completely unexpected.
It's a good question. I mean, my FMD is my day job and has been my day job for all this time. So I think I was the person in Germany probably most worried that FMD would come to Germany. And over the years, I would always like make the rounds, speak to people at like speak to farmers, speak to official veterinarians, etc., and always say, hey, don't forget FMD is still here, because you tend to get distracted with all the other diseases going on. We had a lot of avian influenza, we had a lot of African swine fever and blue tongue, and people were just dealing with all these things. And I was just like, don't forget about the FMD. But at the same time, of course, I hoped that I would never have to deal with it. I thought, well, maybe we have things under control. Maybe people are careful enough in what they do so that it's not going to happen. But I was always very fearful that if it would happen, and maybe we found it too late, it would like this big catastrophe like 2001. And so when that call first came on the Thursday, I mean, the first thing I was worried about is, so I asked, where is the farm? And then when I heard it's far away from FLI, that made me happy, because I mean, when you run an FMD containment lab, and then you hear, oh, there's an outbreak just down the road. That's not good. So it was far away. That made me happy. But then it was just like, okay, we have this first case, and we got the report from the regional lab also with like a mid-20s CT value. So it was clear this is a real thing. This is not contamination. This is not like them misinterpreting the result.
It's
Sure,
not a
we need to
drill.
confirm it, but there was no way that was not real. So we kind of knew from that moment that we had a problem, but we obviously had no idea how big the problem is. So you worry what's going to happen the next few days. Like, people are going out, that hopefully farmers will look at their animals more carefully after they hear the news. And like every minute you just wait for the next phone call and the next and the next, but they didn't come. I mean, we did. So for the first couple of days, we were quite busy in the lab with like the virus characterization, etc. But then when that was all said and done, the real work was done outside, was done by the states, and we were really just sitting there. I mean, we had a lot to do. We had a lot of like phone calls, people asking questions. We had to do risk assessments. We had to do all kinds of policy advice, but we had no actual. I mean, we were just waiting, really anticipating what would happen. And then slowly over the next week or the next 2 weeks, we had one additional suspicion that turned out to be nothing. And then you slowly kind of think, okay, maybe it was really just that one farm. And then you just don't believe it. Why would you have an FMD outbreak that's just one farm? I don't know. It was an interesting time for sure. I mean, looking back, it was good. It was very stressful, but I'm happy that everything went the way it did. We found some things with like our contingency planning and with the procedures that maybe didn't work as well as we thought when we first made the plan. But I mean, that happens always. And overall, I was very happy with the collaboration both within the FLI between the diagnosticians and the epidemiologists, and then the exchanges that we had with the field vets, with the regional labs. It was all very good. It was in that sense, it ultimately was a positive experience.
Right. Oh, thank you. Well, I will continue just with the one question from the audience, and then I will give you to you, Melanie, from Anna Ludi. Hi, Michael. Thanks for the great talk. For the serology testing, what assays did you use, and what did you do with any non-negative results? As we all know that the SP and SP ELISAs aren't 100% specific.
Yeah, that was a big concern of mine. So we only used the serological assays, the commercially available serological assays. So all the NSP testing pretty much was done with the IDVET NSP antibody ELISA, because they have the contract, a contract with Germany for like emergency diagnostic reagents. And then all SP testing initially was done with the PrioCheck type O antibody ELISA, because the IDVET type O at the time was not licensed in Germany. That happened a little bit later on, but in the beginning only the PrioCheck was licensed. And so initially we just tested everything, both we did and the regional labs did, with in terms of serology with the NSP ELISA, and any non-negative results in the NSP ELISA would then be forwarded to us for like retesting. Usually we confirmed the non-negative result in the NSP ELISA, but since then we knew we were dealing with a type O virus, we then took the type O SP ELISA as kind of the confirmatory test, and that was always negative. And with that, we then just said, okay, that's good enough. And I mean, you also look at other results you have from these farms. Like in many cases, these were samples from the zones. So you did not just have the serology, but you also knew that there was a veterinarian on the farm. They also took swabs. And when all that was fine and it was just that serological result, then we didn't do anything else. We left it at the negative SP result. And by and large, it weren't that many. I was really worried that we'd have lots. in particular with the wildlife. I was very worried because we said we're going to test all this wildlife from the containment zone. What if we have lots of positives in the wildlife? What are we going to do with that? But the wildlife was fine. I think by and large, from this initial round of testing that then went in the WOA dossier, we had like 6 non-negative results out of the 10,000 tested. So it wasn't bad at all. And they were all negative in the O, and that was it.
Melanie?
Okay, thank you. Another question from Aldu. Seth, he wants to know if you tested the water of the lake. So if you did any RT-PCR, and if so, did you detect virus? And I think his question is coming out of interest. We've been chatting to them regarding industry wanting us to check, to test a disinfectant for them. And they keen that there's transmission through the water, and they want to now publish this, and they want us to do studies on that. So there's no literature to support it. So what is your take on that?
Yeah, it's a difficult topic. When I first heard about that lake thing, so they came to me and they said, oh, so give us a risk assessment. How likely is it that the lake water is a problem? I also looked through the literature. I didn't find anything. There's one Dutch paper, but it's mostly about like people pouring milk into canals, kind of thing. So it didn't really apply. And I was worried. Well, no. I thought I'm just going to do a calculation. So I knew that this buffalo was dead. So how much virus was in the saliva? How much water is in the lake? How much is it going to dilute? And I just hoped that we would arrive at a figure that was so low that I could say no, but forget it. The numbers didn't really work out that way, so I couldn't really reject it on a scientific basis. But I agree with Aldo that I don't think it's a big deal, and particularly, I mean, what's going to happen? So the worst thing would be sure maybe a deer comes and drinks from the lake, but we were already weeks after the outbreak here. I mean, I didn't believe the whole thing, and so we decided to go with the lowest impact solution that was really just fencing it off and leaving it. But no, I did not get any samples from the lake, and I don't think the regional lab tested the lake either. I'm just going out on a limb here, and I'm going to say I'm 99.999% sure that it would be negative.
But in an endemic area, what would you think the role of water would be? If there's flooding and there's washing of virus in the rivers going to another farm, do you think the risk is high with transmission?
No, it's not. I'm sure it's not high. I'm not sure it's entirely non-existent, but it's certainly not high. We're talking a lot of water, and dilution solves a lot of problems. And then you have, I mean, I don't know about the weather right now in winter in South Africa, but as long as you have like sunlight, etc., I think you have lots of other things that are bigger risks than the water. And it's probably better if these people spent their energy on controlling the things that they can control more easily than worrying too much about the water, is my personal opinion.
Okay, thank you. Mariana, over to you.
Okay, so probably we have time for another question. This is actually a triple question. It's a triple one from Benedicto, but no, I will try to make it short. The death of 30 out of 14 water buffaloes seems really high for adult ruminants. So this could this mortality have escalated to 100% if they were not killed before after that? And this suggests that the immunological naivety of the non-endemic and high biosecurity setting are what are the implications in the FMD risk modeling in such populations? So could be this an element that should be taken into account to make a risk modeling?
So I think the first question is easier to answer. The other buffalo that were still on the farm kind of were past the peak. So I don't think that any of these animals would have died the way these other animals died. I mean, they all had antibody, they all had like fairly low amounts of viral RNA in the samples that we tested. So no, I don't think we would have seen more deaths than the 3 that were already there. But at the same time, I do agree that 3 out of 14 adult animals, I mean, there were 1 or 2 calves among them, but that only makes it worse. It's extremely high, and it's not what you expect in the literature. But maybe water buffaloes are not cows, and I mean there are some reports in the literature from Egypt, I think, that describe severe FMD in water buffalo. And I personally just I know too little about them. We do intend to do an animal experiment with water buffalo and FMD sometime in the future, and maybe we can get a bit more data from that. But right now, I can only report our findings. And I mean, if you really are strict, I don't know whether the first 2 buffalo also died of FMD. I don't have any samples from them. I don't have any description of them that goes beyond they were dead. Who knows? Maybe they died of something else. I don't think they did. I think they died of FMD, but I don't know. And I don't know why all these adult animals died. And I don't. Maybe what would be interesting to know is also to have a little bit more. Well, let's just call them stories from Western Asia, where this virus is circulating, from Iraq, from places like that. Do they see similar mortality in adult animals? I don't know. But if they also do, it may be a feature of the virus. I don't know.
You don't have records on that, so
I personally don't, but maybe somebody in the audience has some knowledge, and I would be very, very curious to hear about that. And in terms of how does that change our approach to risk modeling? That's not my area of expertise. I mean, I frankly don't know. At the end of the day, whether they die or not doesn't maybe change the risk of onward transmission much. In a way, you can say an animal that drops dead maybe is better for outbreak control, because A, people tend to notice it quicker, and B, that animal's not going to shed much virus anymore. I don't know.
Right. Well, there's also a comment from Benedicto that says that the R1 values for all 3 vaccine strains were below the 0.3 threshold. Well, I noticed that there was a difference between the Beringer calculations with 0.6 or something, and the ones from the European lab. But,
I'm not an expert on vaccine matching. I mean, you are way more than me, Mariano. But I mean, what I've been told, or I keep being told, is that this is one of the reasons why Boehringer, for example, likes to move away from the R1 values and really just look at the heterologous titers, which were very high. And the R1 values being as low as they are, to my understanding, just comes from the fact that the homologous titers were extremely high. And then you have outrageous homologous titers, and you have very good heterologous titers, but the R1 values are bad. And that's obviously not helping the interpretation.
Yeah. So, Melanie, I don't know if there are any more questions, or do we have time? It's up to you.
Yeah, I think we've actually 6 minutes over time, so we've run out of time. Unfortunately, there are some questions we have missed. So I think I'll just ask Kayla to collate those questions for you, Michael, if you can just respond on a separate time point for them. And just so the audience knows that we are going to post the webinar on YouTube. There's a chance for you to also post your questions there. We will monitor it and get the answers to you as soon as possible as well. So thank you, Michael, again for being present and for doing this for us. We really appreciate it. Very nice presentation. And thanks everyone for joining us. And until the next time, keep a lookout for our advert for the next presentation. Kayla,
Yes.
over to you to close for us.
Just a little bit. So people are welcome to email me as well, the questions if they have any questions. And also, I'll be at the GFRA meeting in the fall. So if you want to talk FMD,
Awesome.
maybe join us there.
Excellent. Thank you.
Great. Yes, Michael, thank you so much for such a great and engaging presentation. Thank you to everyone who joined today as well. Just a quick note, as you're exiting the webinar, you will see a short survey pop up. If you could just take a minute or two to fill that out, that is really helpful for us as we continue to expand upon and improve this webinar series. Thank you so much, and we look forward to seeing you at the next webinar and hopefully at the scientific meeting in October. All right, thank you all.
Thanks. Bye.